Okay, Dr. Shinoy. Good morning, everyone. I would like to welcome all of you for today's talk, which is organized by MycoAsia. As you may heard that MycoAsia is trying to make a platform between senior mycologists and the other mycology interest people in Asia. So today's we are going to have a hot topic in the mycology at the moment, which will be delivered by Dr. Mubashar Raza. It's on advanced phylogenetic analysis. So I would like to give a brief introduction about Dr. Raza. He is currently working as a postdoctoral fellow at Institute of Microbiology of the Chinese Academy of Science in Beijing, China. Dr. Raza completed his PhD in microbiology from the University of Chinese Academy of Science in 2020. His PhD advisor was a famous plant pathologist, Professor Li Chai. Besides, Dr. Raza was an exchange student of Professor Kevin's lab in Thailand since 2014 to 2015. Dr. Raza is a very important person in Asian mycology because he is one of the pioneer mycologists of the young generation in Pakistan who recently published a fungal list of, of Pakistan. At the same time, he established a website as well. He has been working as a reviewer and editor and a guest editor of several reputed journals, and we are happy to say that he is one of the associated editors of MycoAsia. In China also, he has a good reputation as a fungal uh, mycologist and as well as a pathologist. So he has a good experience on advanced phylogenetic analysis, especially regarding resolving species complex and important emergent pathogens. So I would like to welcome Dr. Raza to deliver his talk on advanced phylogenetic analysis. And maybe Dr. Shenoy, maybe it's better you remind the rules and regulations again, and especially about the quiz that will be held after the presentation. Thank you so much. I hope you will be having a wonderful day.
Thank you, Dr. Nalin, for the kind introduction of Dr. Mubashar Raja. Um, Ladies and gentlemen, before Dr. Raza starts his online training for advanced phylogenetic analysis, I request you to please mute yourself. Don't switch on your video unnecessarily. And at the end of the training program, maybe at 12:45, 12:50, we will have an online quiz or assessment quiz. If you score more than 10 or more, you will be eligible to get the certificate of completion. Thank you very much. Over to you, Dr. Mubashar Raza. Thank you.
Thank you so much, Billy, and thank you so much, Nalin, for my brief introduction. So I would like to start my presentation about the advanced phylogenetic analysis. So the point of the discussion of today's presentation, we will learn about how to construct the phylogenetic tree and how to see taxonomic status of the species and how we can distinguish the intraspecific and interspecific differences between the species. Moreover, how to choose outgrowth, which is very important, as well as what are the similarities and differences of the maximum likelihood, Bayesian method, and other different methods are used for the construction of the phylogenetic tree, as well as how to delete and modify the tree if it is not good in shape, and how we can improve the quality and how we can make it beautiful for our viewers and for the publication. On the other hand, I would like to introduce the book that is very important if you want to learn about the phylogenetic analysis by the G. Hall, Phylogenetic Tree Made Easy. So later on, we will share this book and all this presentation with all our all our participants. On the other hand, if you have any question, directly email to me at my email address, as well as you can have a contact with me on my research kit. And then in the same time, in the background, what are the different softwares are used, and I will and on the other and phylogenetic tree constructions, what are the method? Most importantly, we will use the Roxmail and Biosemin analysis that are mostly used in the publications. We will learn at the last. We have some question answer session, and later on, after the question and session answer, we will practice. We have prepared the online practice session. If there are some questions from the audience or the participant, they can directly ask to me. So the first one is the background knowledge. Okay, then the phylogenetic analysis, why we create the phylogenetic analysis. Today, we all know the phylogenetic trees appear very frequently in the molecular papers, especially in the taxonomy. And the phylogenetic tree is composed of the lines called branches. The branches at that that intersect at the terminate at the node at the tips. The node at the tips of the branches represent the taxa and the two nodes, and then make a clade. Taxa also called as a trait. And the bootstrap values that come from the analysis that we use to construct the phylogenetic tree, called the values, those values called as bootstrap values, and the section from the two taxa separated from each other called node, and the outgroup. We will discuss about the outgroup, how we can choose that, and the length of the evolutionary branch. That number represents the branch length actually. So in the next slide that we will talk about the traditional tree diagram. Traditionally we are using the rectangular tree, and other ways to represent the tree are the straight tree, curved tree, circular tree. That is more common in the especially a microbiome data, radiation tree, and phylogenetic tree. If you have any idea, we can see that they are the rooted tree or unrooted tree. All these trees are unrooted tree. So in the monophyletic groups, paraphyletic groups, and polyphyletic groups. And the monophyletic groups is a taxon that consists of most recent common ancestor, and they have descendants. Monophyletic groups, all the descendants of the ancestor. So if from this point, simply point 7, monophyletic group, if we consider the whole, this is a monophyletic, but they separated from this point that called is node. So the simplest one is the A, then the B is they have the common ancestor, the 2.2. So if we A, B, C are that is one also called is clade and the monophyletic, and overall this clade is we consider as well as the monophyletic, because they share or they consist of the all descendants, and they share the most common ancestor. So in the other hand, the paraphyletic groups is a taxon that consists of more recent common ancestor and some of its descendants, but not the all. And paraphyletic groups does not include all the descendant of the ancestor, while the polyphyletic groups is the taxon that consists of unrelated organisms who are from the different recent common ancestor, and this group from polyphyletic groups lack the most recent common ancestors. And while defining the species, and most of the biologists, while defining the species, they have the species or organism. What is that? There are different species concept originated in that way, and most recent are the morphological species complex, biological species complex, ecological species complex, and the phylogenetic species complex. Morphological species complex consists of the morphological divergence, and it is commonly used criteria for the species complex, and it is in operational and theoretical species complex is a ecological species complex adaptation to the particular ecological niche, and most important the group of the certain area, while the phylogenetic species complex based on the nucleotide divergence time, and these are the smallest group and the lineage. On the other hand, there are more than these species complex. There are 26 more species complex. If someone want to have the details about all these, and can go to this paper, these articles, and all the definition and regarding the information can be found in this paper. But the species are the real, but the classification are not, because the most biologists accept that the species are rare following the course of the completion of the inceptant species, hybrid, sibling species, and the like. The reality of the species is different from that of the chemical elements in many ways, not less in the fact that they are linked by historical lineages of descendants. While constructing the phylogenetic programs, we have, while considering the phylogenetic programs, according to the various authors, species are one of the fundamental unit of biology, making them comparable in the importance of genes, cells, and organisms. Some of the fundamental units at lower level of biological organization, however, because species exist at higher level of the organization, human observing them. So species delimitation from that, which why biologists have symposia devoted to the topic of the species complex. So we use the different phylogenetic programs to what is species and what are the close related species, and phylogenetic where they fall. So in the construction step and the commonly used software of the phylogenetic, we have probably 4 steps for construction of the phylogenetic tree. Among them, and first of all, we identify the protein or DNA sequence of the interest. On the other hand, then identify other sequences that are related to the sequences of the interest, obtain the electronic file of those sequences, whether from the references sequences or from the other publication, and then align those sequences using the resulting of the alignment, generate a phylogenetic tree and construct them by different software like figure tree, other things. So the first step is data preparation and sequence assembly. We use different software, Sequman, ClusterW, BioEdit. All these are the software, and then we use the different online programs, master alignment, muscles in the muscles, Masquerade, G-Block, and then phylogenetic reconstruction, POP, MAFT, as well as we also can use the MAFT software, online software, J-model test. Before constructing the after the alignment, we need to run a phylogenetic J-model test, whether and for different different phylogenetic tree, or that for the different methods. Some we need a J-model test, in some we don't need. And for the species delimitation, I will skip this part, but for we will in the detail we will discuss about the data preparation, sequence assembly, splicing, sequence format conversion, and different format, what the file we need for the required for the different software. At the end, how we can make the phylogenetic tree beautiful. The second part is the phylogenetic reconstruction. First of all, the sequence splicing is very important, because if there is a wrong in the alignment, if we did not choose the reference sequence correctly, it will affect our alignment, and at the last we will obtain the wrong phylogenetic tree. So we use the sequence Sequman DNA star software, First, we open the software, so go to the file, start, click add the sequencing button, and a panel to the add sequence to be supplied. So it is very important when we sequence the when we do the sequencing of an organism and get the result. Most of the time, in our case, we get forward and reverse primer to compare whether our sequence is right or accurate. In most of the cases, in previously the normally if there are funding issues, then the authors or the researcher, they use just only forward or the reverse sequencing. In that case, we are not sure and we cannot compare the forward and reverse to get the contig sequence, whether our sequence is right, accurate or not. So if we have the forward and reverse sequence, we will add both forward and reverse file. When we like as a sequence file, select the from the same organism, we select the sequence file and then add and then open. After that, we get this, we can see the peaks of the sequence that we get from the sequencing company or the sequence. We can see that if the peaks are accurate, we can compare the forward and reverse. And if those the peaks from the most probably from the forward and most probably at the end, from the beginning, from the end, the peaks are not accurate most of the time. And then we can delete those wrong nucleotides to get the more accurate and complete sequence of the forward and the reverse. And then after the sequencing data is check corrected, we click the assemble to assemble the data, and the and contig sequence will up, and we separate that, we save that contig sequence in a FASTA file. And then we have the one sequence from forward and reverse. That's a sequence called as the contig sequence that could be used to align with the reference sequence. Suppose if there is an overlapping between forward and reverse, in most of the cases forward primer, forward sequence is accurate, or sometimes the reverse sequence is accurate and forward reverse sequence is not fine and the peaks are not accurate. In that case, what should we do? And we check, like here the C and here the peaks are not like in the, this is the forward sequence, this is the reverse sequence. So we can check there is a C, here is C is absent, so we can consider it C. We can include it. Otherwise, if we have just only reverse sequence, this nucleotide will be skipped. So this is the importance of the forward and reverse, the both regions, and then it will be more accurate contig sequence. After that, we blast that sequence in NCBI to check whether the NCBI results match with the which genus, which species, which organism it most close related to. That is very critical steps. Most of the, if in case of the ITS tell us the most probably at the genus level, sometime at the species level. So we can check from the reference sequence what sequence we have to choose. Most publication, most publication they can see they have provided the type specimen. So if the type specimen is not, we can't find in the publication or in the reference papers. So we have to check carefully here the blast result, the sequence from the type material. We also need to check the authors, and we also check that the sequence is accurate, because in NCBI most of the data is wrongly submitted, and but with the help of the taxonomist now they are trying to improve the NCBI database, then the more accurate to get the more accurate blast search. So that is just an example how we can get and save the references sequence, and then here we blast and then check the 99% identity and click on this. Get the FASTA file to the gene bank. We can save or copy, and we can download the FASTA file of the reference sequence to align with the to align with our sequenced strain or sequence organisms. In the blast search, the user should be that I have highlighted that caution before using the data. Check the validity of the sequence and compare any information like gene bank session number, culture code, species name, depositor name, whether the depositor are the real taxonomists or just deposited by the plant pathologist. Because if it is deposited from the taxonomist, most probably the sequence will be accurate. But sometimes there are still some mistakes when we are analyzing the data. We can find, like in case of when we are dealing with the big data set, the LSU can be named with the SSU, so mistakes happen. So we have to cross check the material sequencing. We cannot only rely all the database from the NCBI. So this is all the references sequence, how we need to arrange the reference file for run the phylogenetic tree. For that will be we will see. But here we first of all we have to collect the references sequence in one file to obtain whether all the species, all the references sequence are in the one file with our sequence strains. Mostly that sequence should provided that's the important of the gene bank session number that provided in most of the publication or in the supplementary data of the publication. We can also get the all information from the ITS, LSSU, RPB2 or the other gene region that are important for the so we don't, in that case, if we know which genus or which order or family our isolates belongs to, or the blast search are telling to us, so we can check the references papers or reference articles to get the session number or the reference data from that publication. That will be more easy and accurate. So in the most of the part we consider the rooted tree, unrooted tree, and what kind of information they tell us. A tree is said to be rooted if there is a particular node, the root at the bottom from which are unique directional path leads to each extant taxon. So in the rooted tree, that tell us about the order of the descendant from the rooted towards the tip of the tree. But in the unrooted tree, that tell us nothing about the direction of the evolution or such as the order of the descendant. But unrooted tree tell us about the phylogenetic relationship. On the other hand, the midpoint rooting tree places the root at the middle of the longest path, the two mostly distinctly taxa. But the wrong selection of the outgroup influence the phylogeny reconstruction, such as the different topology. So outgroup selection is very important. So while choosing the outgroup selection, an appropriate outgroup is the sister group that are sequence that are neither phylogenetic close or phylogenetic distinct from the in groups. In case of the travel phylogenetics, such as species delimitation and the resolving species in set of sequence at the genus level, a proper outgroup is chosen from the sister group rather than the less close related taxa. In case of the higher level taxonomy, like in the order level, so when we are constructing the tree of the different families, so the most probably we should choose the closely related family or from the different order and close related family. So that's why, and or if we still don't know which outgroup we have to choose, we can check the reference publication and check now it is very easy because of the references articles and publications are available. We can check from them, and close related and sister groups we can choose the outgroup and extract the sequence data from the NCBI and construct the phylogenetic tree. And then after the collection of the sequence data and the reference sequence data, choose of the outgroup, we have to align the reference sequence with our isolated strain of the sequence. So most of the time we use the MAFTA alignment, that multiple alignment program for amino acid and nucleotide sequence, is in that case there is arrangement of the sequence to identify region of the similarity, where the main object of the alignment is to create the most efficient statement of initial homology, as different sequence alignment method affect phylogenetic topology. In resulting, the phylogenetic is highly dependent on mode of the alignment, which which include pairwise alignment and multiple alignment rather than of the methods of the phylogenetic reconstruction. So this is the online tool, MACTA alignment. We can just go to the website and we can put our data here, or we can upload the file and choose some default program, or it depends on our what kind of data set we have. So we choose the program, and we can see the results here. If there is a blue line, red lines indicate that our sequence are fine, and they are in the forward direction. All the sequence are in the forward direction. But if there are blue lines, it means those few of the sequences are in the different direction. So we have to back to our file and make them in the right direction with other references sequence, and then need to align again. And we, after the alignment, we can download the FASTA file. And how this software alignment works in the background, we at the front view, we just use some options and how it is working. This is a background information, and all these information are more information are available on the online website. They provided in detail. And the progressive method is incremental method, in which the 100 cycles are and included the maximum 100 cycles, and output file is and maximum 100 input files. And then other one is an iterative refinement method with the iterative optimization and iterative refinement method using the both the WSFP and consistency scores. While we are doing the practice session, we will see that all these in details. So here I don't have the animation. Actually, on this slide is the animation and the background, how it is working. For example, let's see session one, and let's the last one. This is the reference sequence input. For example, in the reference sequence, they have the for IT, they have sequence the ITS1 and ITS1 region by using the ITS1 and ITS2 primer. But when from our isolates, we have used ITS1 and ITS4 primer. In that case, we have a long region. But when we align with the reference sequence, so this is the extra gene region from ITS2 to ITS4. When we will align, that part will be removed. So we will have the same and forward same sequence length, almost most probably for 550, 400, 420 of the ITS length we can get. So extra part we need to remove. So in this is how the MACTA alignment works in the background. And after the alignment and the phylogenetic tree is constructed, and we can use the MAFTA alignment, but most of the time just to see the initial or preliminary results, to see that whether our alignment is okay or not. Just MAFTA alignment, we cannot rely 100% on the MAFTA alignment, but we have to see the visual observation, your experience. Our experiences matter a lot to see the alignment is all right or not. If there are some mistakes, we still have to do some correction at some points after the alignments. If that is still there are some issues in the MAFTA alignment, we need to, there are more conserved regions or more conserved side in the alignment. So we have to use the G block to facilitate the next evolutionary analysis. In particularly, sequence with the relatively large differences have many gaps region after the alignment further need to process with the G block. This is also an online portal where we can do the further alignment for the processing. So After the alignment, we have to choose the model selection. Model selection, the commonly used software are the model test, Mr. Model test, J model test, and so on. So in the model test, 56 DNA surrogate models are present, while in the Mr. Model test contain 24 models, and in the J model test contain total 88 models. So we need the FASTA file that we have multiple file, single loci FASTA file or the multiple loci FASTA file. We have to load the DNA sequence. When we are opening the J model test, load DNA sequence, and when it is loaded, then go to analysis, compute likelihood score, and then run. After the running, it will take some time, or sometime if your data set is very large and very big, it will take a long time. So we need a high processing CPU computers for this analysis. And normally we cannot run on our laptop, but we have some desktop computers for it to run. And after running, it will take some time, and it finish do the ACI calculations to get more accurate, or more accurate, as well as the correct model for the for our further analysis. So it will choose from the 80, because we run the J model test. So in the J model there are total 88. So it will choose one, the best one model for our analysis based on our sequence data. After choosing, after choosing the database, we have different softwares like the neighbor joining methods, minimum evolution method, and maximum parsimony, maximum likelihood, Bayesian analysis. And so in the publication, which method we most of the time we are using it to represent our data, we present our phylogenetic tree, is the maximum likelihood, that is ML tree. And then on the ML tree, we have two values that is generated, bootstrap values generated from the ML tree, as well as the Bayesian interference. So why these both values are present on the phylogenetic tree? Because it compares the topology, whether the tree generated from the both methods are right, their topology is accurate or same. So the values representing the same topology of the both, but in the publication we use maximum likelihood, the tree that is generated from the maximum likelihood, that is because it is more suitable for the evolution. The result obtained by maximum likelihood methods are better, and for the sequence closely related species, maximum parsimony, and the sequence of the distant species neighbor joining method and maximum likelihood methods are generally used. So in the details we will discuss about the maximum likelihood method and Bayesian analysis. On the other hand, we also what are the differences, what are the basic features, application, advantages and disadvantages of all these methods. So in the neighbor joining method, this is based on the principle of minimum evolution, when the merging classes, not only the classes to be merged are required to be close, but this application is based on the evolutionary distances sequences, but short sequences with small evolutionary distance. But the disadvantage is that all the sites on the sequence are treated equally when we run this neighbor joining method. On maximum parsimony, no substitution model is required for it. So if for that maximum parsimony, we don't need to run the any model test. But the application or the limitation of this parsimony is the closely related species sequences, number of sequences should be the less than 12. There are few residual differences in that case. So a large variety of sequences will be attracted by long branches, leading to the incorrect phylogenetic tree construction in maximum parsimony. So why the maximum likelihood method used in the publication? Because specific alternative models to analyze are given to set of the sequence data, and application is best fit for the evolutionary fact, and it is very good statistical foundation. The likelihood method can obtain the minimum variance of the parameter statistic when the sample is very large, and ML method, maximum likelihood method, is the best tree building algorithm in the line with the evolutionary facts. So with maximum likelihood, with compare with the comparison with the Bayesian and Mr. Bayes, because no bootstrap test is required in the Mr. Bayes, large and complex data is also applicable. We can use how large is data, it does not matter. So it also, we can run the Bayesian Mr. Bayes. We can have the solid mathematical and statistical foundation to handle the complex and near realistic evolutionary models. But the limitation of the Mr. Bayes is the sensitive to the evolutionary model, the posterior probability of each amino acid specified in the Bayesian interference method established under many assumptions that may not hold in reality. So what are the common software that use for the maximum likelihood and Bayesian analysis? So for the maximum likelihood, a lot of softwares are available, but it does not mean that I have not mentioned here some of the widely distributed. Maybe my failure to mention a program does not imply that program is not useful or valuable. I have chosen to include a minimum set of programs that will allow to implement the methods I discuss in here. So maximum likelihood, we can use Philip, Paul, but we are going to use for the command-based Ruxmail method. We are going to use the Ruxmail method to construct the maximum likelihood method with comparison with the Mr. Bayes to construct the Bayesian inference method. Still, we can, if someone else, they are very familiar with other software, they can use and compare the topology of the two. But in the publication, in the last, so last, we have to provide all the information, which method we use, which model test we have choose for that. We have to provide all the information. So if someone else or the reader, they want to construct the phylogenetic tree by using the same method, they can construct and they can get the same results. So what are the requirement or the file requirement, because we cannot use the FASTA file for all the software. Some software, they require Philip file, some require FASTA file, some require Nexus file. For the maximum likelihood, they require FASTA file and Philip file, both are okay, but for the Bayesian analysis, we use the Nexus file. So first of all, we have to convert the FASTA file that we got from the MAPTA alignment. We have to convert by another software, or maybe online software. We have to convert the file into the Nexus file for the Bayesian, or if it is already in FASTA or Philip, we can use for the maximum likelihood method. So the professional software recommended for this software is marked in the red. So for the Ruxmail, Philip ML, IQ tree, for the Bayesian, Mr. Bayes or PhyloSuite, both are okay to construct the phylogenetic tree. But here today we are going to discuss the Ruxmail in detail and the Mr. Bayes for the Bayesian analysis. But we have to consider and to note that if we have constructed the FASTA file, that FASTA file should not contain any sequence, any space, tab, new lines like commas, brackets, or something like this. If there are any dot, any comma, any semicolon like this in the FASTA file, there will be error in the construction of the phylogenetic tree that we cannot process. In that case, we have to see our all file again to see whether where is the mistake. So there cannot be sequence with the same name. We cannot repeat the same name. Sequence could be same, but we cannot use the same name or repeat the same name. We have to use some keywords to replace the name, whether they would not be. If they are the same, there the system will stop the construction of the phylogenetic tree, or maybe we will get the error. Another point to be noted that the sequence cannot be completely composed of the unknown symbol, and there are other, if there are question mark, if there is X or the asterisk mark, then still there will be an error. But the other DNA sequence can consider that N, O, or other to separate the genes. If there is a single loci sequence alignment, that is okay, but if it is a multi-locus alignment, then we have to separate that alignment with the different or alphabets, like we can use N, 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 or O, O, O. We will see further in our alignment. So for the Ruxmill, we have this is for the phylogenetic tree construction. We have this command to construct the phylogenetic tree, but on the backup we have some files. But the main point is this is a command, and how we can read that command and how we can change the information in this command. So Ruxmill HPLC PTS, what is according to the computing speed of the CPU. So Linux, if we are running this the phylogenetic maximum likelihood method on our Linux, they have 3 versions: on standard version, SSE 3, AVX version. There are 4 versions for parallelization, sequential, and this is just all about the CPU or the system requirement to run the phylogenetic tree of the Ruxmill tree. And S is the input file format. We can, after the S, we can give the input name of the file. We have to mention that, for example, if our final alignment or multi-locus alignment name is AAA or BBB, so we have to write here the file name here, and after the N is the ZZJ is the output file name. So after the completion, we will get the output file name with the same name that we have mentioned here on this point. This is a model for the Ruxmill that before running we have which J model test on our database that we got the result, and a nucleotide or amino acid substitution model, a mutation rate model that we got from the running the J model test or Mr. model test, and T40 is equal to the 40 threads are premise that is server supports only limited to the multi-thread version of Ruxmill, and is the number of runs of different starting tree. We have to, for Ruxmill, we have to run, it will run all about 100 times, and after running the 100 times, we will get the bootstrap value, for example, and 98%, 99%. It means that after running the 100,000 times, after running the 1,000 times, the tree or the bootstrap or the topology would be 98% similar. Like so, in that case, we can read the information on the phylogenetic tree from the bootstrap values. And this means that P specify a set of random numbers. So most probable time randomly we use 1, 2, 3, 4, 5 random numbers, any random numbers we can choose. And after that, FA is a rapid bootstrap analysis, which can quickly perform bootstrap analysis to find the maximum likelihood tree, and specify the last one, the digit, specify an integral random number, establish the faster bootstrapping, unlike the previous version of the Ruxmill. And after completion of the Ruxmill tree, we will get this by Ruxmill bipartition branch label, or most probably we will have the 5 files generated after the completion of Ruxmill, which is an optimum. So this is our final file that we will use to use, such as figure tree or the software to use to construct to see the result of the phylogenetic analysis, and it will have the bootstrap values and branch length information. And not only roxamyl nucleotide, not only the GTR models, but there are but others nucleotide substitution models, several combination of mutation rate models are fellows, but so sometimes not only GTR models is we can get, but if there is something as the best model, that are the information here. So don't be say what is the model and what this. So they are here are the some information I have provided to see that we can use and what does that model means. And the second one is the Mr. Base and Mr. Bayesian inference of the phylogeny. And there are two for the 32-bit system is separate file and 64 system is the separate file. So we can download from this link. And I mostly use 3.2 to fix a number of bugs because the previous so updated version are also available that was almost in 2018, but this 3.22 is about in 2000 was released in 2022. So the program Mr. Base performed Bayesian inference of the phylogeny using the variant of MCMC like Markov chain protocol to approximate the posterior probability of the tree. But Mr. Base has a command line, same like maximum likelihood, but in case of the Mr. Base, the command is bigger than the Mr. Base. So, but that is easy as a command. So that command actually we need to use the nexus file. First of all, let's go here. So how we can there are okay. This is mega software. We can upload the file. After the upload, go to data export file and convert it to the nexus. After the nexus, we will get the file. In this way, there are the nexus file title, begun data, but there are some information we have to remove or we have to replace or we have to update this file information. Like the final file, we have to remove that title, begun text, we have to say that begun data. So dimension NTXS is equal to 26. So here these are the information, but at the last, so from this area, from this part we will remove, and this information will be connected to this. So we have to generate the file, the nexus file in this way, like a dimension NTS is equal to 20, whatever the value would be. So different files every time the different values. At the last, this is a command line for the Mr. Base. At the last, we can update the information in this command line. We have to provide the model test after performing the Akai information criterion, like the best model, they selected the GTRIG. So in this model, the frequencies is the 0.02 different frequencies like A and C, then the A and C, how many times, and the information, this is bioinformatic analysis there after. So we can use this information here, and P inverse values here, P inverse values, and the gamma shape values is here. So we can change this information in the command line, and after that we can use the software or some files in the backup. For sure, we will provide you all the information and the software to run the command line and follow these steps. In this information or in this presentation, we just discuss only or I presented only two. But if there are like Roxmail or the POP or Mega software or other software, if you want to learn about that, there is a book chapter. In this book chapter, we have discussed the like MassQuit, how to use the MassQuit to convert the file, POP software, how to construct the phylogenetic tree, and how to what are the steps to construction of the phylogenetic tree, as well as to choose the. So in this book chapter, all the information and in more detail, the step-by-step information with the figures and step-by-step information are provided. And before you can, all the audience, if they want to learn, they can use this book chapter that published in a book, mycology and molecular biology of the plant pathogen interaction that edited by the Manohar. So this we are going to move the practice session after the break, that how we use, if we have sequence information or the sequence forward and reverse, how we will generate the contig sequence, how we will blast, how we will get the reference sequence, how we make the alignment. All these process we are going to discuss again in the present practice session. If you have any question, please, you are welcome.
Thank you, Dr. Raza. Audience, if you have any questions, please raise your hand and ask. Yeah, Ms. Pushpavati, please.
Good morning, sir. Sir, can you hear me? Sir?
Yeah, I can hear you.
Yeah. Sir, thank you, sir. Nice presentation. Sir, I'm asking about the sequence, processing of sequence. Sir, when we usually process a sequence in forward and reverse sequence, some sequences are like, some sequences are different. Like sometimes A comes in the forward sequence, and the complementary to that G or C comes. At that time, what to do? Even though the sequence, the peaks in the chromatogram are very good.
Peaks in the chromatogram are very good, but sometimes you mean that they are not matched with the reference sequence?
Not with reference sequences. Our sequence itself, both the reverse and forward sequence are not matching in this case. Even though the chromatogram was very good, all the peaks in the chromatogram are
you are right.
peaks.
I understand. Sometimes you mean that sometimes forward and reverse sequences, but their peaks, when you open separately, their peaks are accurate even in the forward or even in the reverse. But when you are going to combine or make the contig sequence, you cannot make them, right?
Yes, sir. I face such a problem with my.
So sometimes this case happens. You are using one software, so there is an other software you can use. Another software, like I use Sigman, so sometimes Sigmans cannot build the or combine them, make the contig sequence. So I use another software, and in that software that is okay. So we cannot rely always on one software. You can use another software to make them if you see the peaks are accurate.
Okay, you use different softwares. So then, which is the best software to choose to see the chromatogram and to build a
BioEdit is okay. BioEdit is okay to see the chromatogram, and for the contig sequence, Sigman is I most probably I prefer Sigman. SEQMAN.
Okay, sir.
Yeah.
Thank you, sir.
Welcome.
Sir, one more request. Hello.
Yeah.
Sir, can you show the slide before the J model test? You are showing that. I just missed that slide presentation. Before the J model test.
This one?
No, sir. No, sir. Just before the J model test. Yeah. Sir, back. This way. Okay, sir. Sir, I didn't understand this. Can you please explain?
After the alignment, in the MAFTA alignment, there are some gaps. Like here is the here. In
Yes,
MAFTA alignment, when we, if I can say here, see here, this is a web page of the MAFTA alignment. we can upload the sequence or paste the sequence, or we can upload the file here. And there is a default function of this website. You can use the default function as well to align your sequence or the database, but it depends on your data set, what and purpose of the study, and you are using the protein coding gene or non-protein coding genes, what is database is, or it is the multiple alignment. Of course, multiple alignment you have to use later, but first of all, single loci, you have to align each loci separately to align them, and then you can choose the information here, the options here. When I will we will do the practice, we will discuss in detail which option and which kind of database we have. So in that case, you can upload your sequence and choose the amino acid. Here I can see the same as input, and below there are options like these option, which option you have to use. Like if you have non-coding genes, you can use like ITS, and which option you have to use, you can use NES and how many number of sequence, if less than 200 or more than 200. And if it is protein coding genes, they have gaps, right? So if there are gaps in if there are gaps are present in that, so we will choose that option on depending on our database, and it will give us the result more accurately. If you will use the default option, ultimately your alignment is wrong. You will get the wrong alignment, and after the wrong alignment, you will get the wrong result. Like this is the step-by-step information. Previously, like I can say, like those plant pathologists, like they have no idea about the taxonomy. They consider like use the only the mega software to construct the phylogenetic tree, and later on they say, oh, why our sequence are not aligned with that, but blast search are showing these results. And they're of course, the very important part is the alignment, and we have to choose each and every step very carefully at this step.
Okay, sir. Okay, sir. Thank you, sir.
So I have a question like, what are the softwares which are very relevant for the phylogenetic construction of the fungi, other than maximum likelihood, maximum parsimony, and Mr. Bayes? Can you hear me?
Yeah, I can hear you. Yeah, there are so there we have three methods I have discussed. Like, so which tree is much better? So we follow the maximum likelihood is more accurate, and then Mr. Bayes, and then Mr. Parsimony. These three others methods. Of course, there are other methods, but they are not much accurate. They cannot give you the exact topology or the more accurate phylogenetic tree.
Because the softwares are keep on upgrading day by day. So there are the upgraded software of Mr. Bayes, like Qualities Beast. So there are many versions of the Beast. So shall we continue with only Mr. Bayes, or shall we go for Beast for the betterment, or it is fine for
Beast is can go for the Mr. Beast, but that is used for for like species delimitation. For that I have skipped that part. I did not include it for the species delimitation, and further for Mr. Beast, and then we can use R as well. So of course we can use.
Thank you very much.
Dr. Raza, please go ahead with your practical session. If the audience still has some question, I think some of them will be clarified during the practical session, and maybe at the end of the practical session, we can have one more question and answer session. How about that, Dr. Raza?
Okay.
Yeah, please go ahead with your practical session. Thank you.
So we have LSU, SSU, and TEF. First of all, we are going to, let's see, this is a single loci, LSU, SSU, and TF. Let's see how the alignment, because these are already aligned sequence. And but see here in the down at the bottom of this alignment, we have this sequence which is not aligned. So we are going to use the, for example, this is a sequence from the from our isolate. So we have added in this alignment, or maybe these in the reference sequences. So we will align them firstly. So 23 LSU by using the marked alignment. LSU, same as input, because in our input file, all the alphabets are in the capital letters, but if we will choose it's not same as input, so in the result or in the output file, we will get the lowercase. And align, output order would be the align. So this is, you can see the LSU file, and we can use, because this is protein coding genes, we can use LS and less than 200 sequence with one conserved domain and the long gaps. And after all, like strong metrics for the nucleotide sequence, switch to, and when aligning closely related DNA sequence, because they are from the same and single loci, they are closely related to each other, we can choose 1PM K2X, that is switch to 1 kbs when aligning closely related DNA sequences. And other option, that will be the default, and we can submit and see the results. So everything is fine because all the sequence are in the forward direction. But let's see, we can choose, I am going to change the direction of this sequence, like go to the sequence and for nucleic acid, go and reverse complement. So it's the direction of this sequence has been changed, and I save it now, save the alignment and close it and see the results now. Here, I will go back here, and all the information would be the same, and uploaded the same file just by single change by, and go to submit. You see, I changed the direction. So one sequence is in a wrong direction. So which one is that sequence? I will open all plots here. It will give us the file. We can see where is the position and where is that. Actually, I know that is the last one. And I just, yeah. So this is a position 64 sequence. I will open that one again. Open file. Yeah, 64, go there. Yeah, see the direction. This is at the 64 number, Astrobibium, and I'm going to change the direction, go to become nucleic acid, change it, reverse complement, and then save it. So I will now again close it and go to back. So in this case, we can check our where are the mistakes, and we can change the direction of the sequences, or even the references sequence open and submit. Oh, I did not save that. Sorry. Yeah, it's fine now. We can download the FASTA format or the result file or alignment file from here. Click on FASTA format and download it. So this is our file. Copy or Control X. I can go to the same folder where I have the information. So this is the LSU in the name. So I can give about like 25, 26, 26, and this is alignment file, right? For in case of LSU, let's see it now. So because our sequence have the large length that we included in this sequence, and this one is the, and we can delete, select from here and then delete. Similarly here you see this is an N, so we can see there is no gap in the alignment, and this is seems okay. And now we can save it, but we can similarly, let's see, let's finish it. I am going to open this file, alignment file with the, I want to show that you see in this alignment file we have said that there should be not comma, no anything. We have separated with the dash, and all the sequences there should there should be a not a repeated name with the same thing. If there is a repeated name or the same thing, then we can add like 1, 2, 3 as much you can to change the name. And you can see and observe the file. This is the, there is nothing, but later on we can change. But of course there are different methods like Excel file. They use different ways to save the time. And in the Excel file that is more easy and more like save your time. But at the initial step, I just need to understand we can use this method. And this is also okay. This was SSU. So let's see. TF. So ITS, let's see, TF also 200, then auto, same.
Raza, your voice is breaking, please.
So we can check the SSU results, alignment sequence results, and see. Oh, there are like maybe we have, if this is a case like and in the final alignment, we can go back and check this original sequence, as well as if it is a reference sequence, we can check and compare with the reference sequence, and we can reduce the chances of the mistakes. But most probably there is a chance that there would be G, here would be G, here would be G, because all we can see in the alignment file here is a G. So maybe we can change it. This is just based on my experience. Well, if we don't have any experience, we should not change any nucleotide. We cannot change and insert and delete anything. Like G. Yeah, no gap. Alignment is okay. See, so the now here is the empty box. What does it mean? It means that the initial sequence with the that was on the top, that is blank. So it is all with the blank. So if it is this case, so we can change the position from this the second one, which having the sequence or the align sorry, the sequence data. We can move it from the up and then do. Then there will be not that kind of case like blank boxes. If we will check and see these results, it means that the empty boxes, it means that those sequence, they don't have any sequence data. Like if at the first position no sequence, and you see all that, because that first one was the no sequence, so they align with that. So we will go back and open this one, TEF, and change it. You see this sequence, let me move it down here. Here now at the top we have the, why it is empty, because in the reference data we have the missing data. There is no data available. Molecular data is available like SSU TEF for this species. So I have saved it again by changing the position. Go back again, and so how many genes we have? We have to do align each and every loci sequence alignment separately, and then later on we should combine them in one file. And how are we going to combine them in a bioedit? Of course, we can use different software to combine them, but make sure that there should be not overlapping of any sequence with other strains, because let's in the next step we will see how we can confirm the sequence are matched with each sequence is matched with each other. Still, yeah, you see now we don't have any gap, but in the previous file every box was empty. So this is okay. If there is a blue one, or in the reverse nucleotide, that should be the blue line. You can download it. The result FOSTA file. Go here. So step by step, each file by these number 23, 24, and that will be not overlapping. I know that which file is a updated version and what is that. So this is already combined file, but let's see how we're going to combine these LSU, SSU, and TF in the same bioedit version. Let's open LSU and here edit. Make sure that all the sequence with the same file, and this is the manual way how to we gonna combine the different loci in a one file. So this is SSU. I'm going to open all the windows in one file. Of course, this is time consuming, but if there is another way, maybe I have a chance to learn as well. TEF. I open, but in this alignment case, each and every sequence is not in the same file. I mean in the same order. Like if you see the first one is acriformes, but the last one very morris, and these are the different species. If I will gonna combine, of course the sequence will be overlapped with each other, and the topology will be the different. So go select the first file, go to edit sequence, the sort out with the name or the title. So all these are with the same title. This one, because I know that the name already I have choose are in the previous step, all the names are same. So if I will sort out them with the same name, though they will arrange in the order. Go to here, sort out, and by title. So I can confirm that, like if I'm gonna cross check with this, can see. Of course, at the last step, I have already checked this one. So if you will see any sequence, this one 34, 34, 34, the same name, the same sequence. It means, for example, just randomly I choose this one, and then 8008, so 64, 64, and 64. And it's mean there is something MFLUCC, yeah, this is all right. And 959595, the last one 3434. It means they are in the same, they are sort out in the same order. And of course here, and just randomly choose this one 34 and 99 CBS Corsi, and this is the same strain isolate and Carvisporus. Just randomly check if there is a mistake, and then we have to sort out that one again. At the last Thalidiacum, at the last here Thalidiacum, at the last is Thalidiacum. So this is all right. There is no problem with the name, so I can combine them with the. And how we gonna combine? Like, okay, in the last, go to like edit, insert, go to top. So I'm going to save this file, save as FASTA file, and this is now I'm going to give the name, like the first one will be the LSU, SSU, and TEF. Like the file name is 29. So the new file, because already with the same name, I will gonna give it 30, LSU, SSU, and TEF combine all, and I can remove the FASTA format. Okay, well, fine. And let's open another file because in this file I have, if the, because I want to separate it each loci by different by n adding. So we can use the command Control C. We can select from here to here, Control C, go to this file, select here by now, and Control Shift V. So we cannot use Control V. We have to use Control Shift V. What will happen? Let's see. Now I have done the Control Z. What will happen if I will just do Control V? And you can see that everything will be n in the horizontal place. But if we do that like Control Shift V, that will be in this order. Because there were the 64 in that, in this file, I just want to copy. I just copy from here. Otherwise, I have to add manually. And here, this is now this I already copied, and then I want to copy this one, select from the beginning, go to the sequence, select to the end, and copy the same command, Control Shift V. It will be down here, the same alignment. So I'm going to add the third one, the third loci, Control Copy, go at the last, and add Control Shift V. This will be here, and now the TEF. Here, I'm going to select from here the same step, add it, select to the end, Control Copy, come here, Control Shift V, and these all the and now they are combined, or after the alignment, that is a multi-locus alignment that we have, and we are going to use for the J model test now. Now I'm going to save it, and then if we want to, because this file is sort out with the same name, I can save it. This one I also can save it, and you can see. But make sure that in this alignment, not that each single sequence should be the empty. If there is empty, that will be wrong, because here, okay, missing data for this species. No, we have that. Okay.
Dr. Raza.
Like, yes.
So now we have reached one stage where we have multi-locus alignment with us, right?
Yes.
How about we take a couple of questions from the audience, just to feel them involved.
Okay.
We are going to take a couple of questions from the audience. If you have any questions about the, Okay, Rajesh, please.
Dr. Raza, is there
Yes.
any particular pattern for arrangement of genes in the database, like a data set?
We can use like the same, like the gene regions are kind of like SSU, ITS, and LSU. We can use the same, but if we are randomly choose, but professionally we follow the same like the gene region, we can follow that one. But if not, that is also fine.
What is the reason behind that?
Because the same region, the reason, if we are using the same order that present in the gene regions, then of course the topology or the more accurate data set professionally. But if it is in this way that I organize, I randomly LSU, SSU, and TEF, and I think it will not affect that much. There will be, yeah.
Thank you, Dr. Rajesh. Thank you, Raza. One more questions we'll take up. Do you have any more questions about the combining the data sets?
Yes, sir. I have a question.
Niranjan, ask, please. Yeah.
Yes, sir. Thank you, Rajesh, sir. I have in addition to the previous question, that as you said, it is right, according to the gene regions that we can arrange, like the SSU, ITS, and LSU. This is the right order for that. So, is there any chance to change, for example, within these three gene regions, if I have a number of sequences, or number of sequences, if I have more sequences in SSU than LSU, should I arrange the maximum number of sequences in the first one, or the lower one is the second? Is that any way to arrange in that way?
Of course, the sequence should be in the all three files. The sequence are the number of sequence, like total sequences here. If you see my cursor, the 64. In all the three files, there are 64 sequences. If there is any missing data, like in the first one was the LSU, but in this case, there is a lot of missing data in case of SSU. You see, this is the missing data, but for completion of the sequence number or arrangement, I included the gaps here because we don't have the molecular data.
Exactly. So I meant to say that if there are many gaps, what my question is that among the three gene regions, should we arrange the sequences where less number of gaps? One is the first priority, and then again accordingly. Is that a right way to arrange? Do you got my question?
You mean that what I got is you mean that if the number of sequences, the nucleotide bases in one gene region is complete, but in another gene region they have the missing data. So what we have to do, right?
Exactly. So if, for example, here total 64 sequences. So
Yeah.
if suppose if I have a LSU region, if I have a 64 sequences, let's say,
Yes.
if in the
Yeah.
case of SSU, I have only 50 sequences.
Yes.
So then shall I go for first LSU, then SSU? Is that
The first, then where you have the missing data, you have to complete the 64 sequences before combining the multi-locus alignment. Because before I have the already filled that gaps and I have provided the same name and used the sequence and provided the gaps in the alignment. So that's why I don't have the I just want to save the time for the presentation. Otherwise, you have to complete the number of sequence in all the files should be the same. Otherwise, when you are going to combine and sort out by this, like sort, go to the by title, of course they will overlap. So when you will copy, you will overlap the sequences, another strain with another strain, another strain with another strain. So that will be wrong.
Yeah, no,
Okay.
exactly. I got the information. I just have a
Niranjan,
little confirmation that the uh,
Niranjan,
I
can I
mean,
get in?
yes, sir. Yes, sir.
I
You
think
can. You
the
can.
question was whether to use LSU, which has got 64 sequences, or whether to go with SSU, which has got 54, right?
Yeah, means I'm not considering the gaps. Only.
No, wait. Whether gaps or not, you have to have 64 labels in all the individual data sets.
Yeah,
Yes.
exactly, sir.
That's for sure. Okay.
Yes,
Maybe you can have this discussion separately with Dr. Raza later. Niranjan, how about that?
yes,
It's a
yes, sir. Sure, sir.
very important
Okay.
question, but maybe we will let Dr. Raza to continue with this practical session now.
Okay, sir. Thank you.
Thank you, Niranjan.
Yeah.
Yeah, Raza,
So
please.
so we have this the final multi-locus alignment, and we have saved it. And after that, let's run the J model test. We are because this one is the 30 file, okay, and this one was the previous file 29. So I'm going to run the J model test. Okay. So J model test. Go to the file. Though this is, oh no. File load DNA alignment. We have multi-locus alignment. It's 30. I have loaded the sequence. Analysis, compute likelihood scores. We can choose model filtration, but it will take long time. And of course, the best model we want and best frequencies, rate elevation, and compute. And then it will start. When it will finish, okay, so we will get the result in a way like so when it will finish, it will show the result like this. I have copied and saved it separately. So I will close it most probably. Because this is working, so I'm going to just want to show how it will work. So I'm going to cancel the program. We have the result here. This is the AI, like you see AKI information criteria. So tree for the best AIC model, and for this one they have choose model, selected this model, and these all the information we have, and we can run after these information we can run the Roxmail tree, and here, sorry. So the 30 we have these four file in the backup, and how the first one is the ITS, and so how long is the LSU, SSU, and TEF we have to see here in the file. Okay, let's see how it gets separated. So 99, 99, yeah, so 995. Or maybe I can just go here till 99. So from 1 to 999 is SSU. So many tabs. So 1 to 199 is LSU. SSU is about 1000 to 1090, 956. Yeah, 956, and then started from 7 to 2676. We can confirm 267. 2675, yeah, is 76. 2676. Yeah, these information are same, and so we can save it because that is same. This is a command line for the Roxmail. So we have this Roxmail exe, the random region 1, 2, 3, 4, 5,000 times. This is a model. We can see information in the model. Here is, yeah, team. Change the model here. Which one? PIM 2 plus.
Raza, your voice is breaking, Raza.
Yeah.
Please.
Okay, now it's okay.
Yeah, it's very good now. Thank you.
Okay, so this is the output file 29. So I can give the name here. Let's suppose 30, no, input file, and it is in the FLF format, or the which format it is. Let's see. Yeah. So this file is in the FASTA format. We have to convert this file to the, first of all, in the Philip, because we are going to run the Rocksmith. And for that, we have the MycoAsia software to convert the file into Philip format, and go to MycoAsia. For every step, we have to use the different software, or maybe if you are using one software, that is okay, if that can perform all the functions. Open file. Yeah, first of all, it will save in the Philip DNA. Philip DNA, because this is the nucleotides, and then sequential matrix. Stop. Why? Let's see. I tell there. Yeah, now this is okay, Philip file. But this is the 29 I converted. But I have to convert the 30. Let's see. Okay, so this is a file conversion way. So Philip file, I can use the and give the 31. And then after that, it was converted into Philip, and then file export. It will be the Nexus file. DNA Philip, that was. So these are the number of taxon name. If I will select the name 10, and our number taxon names are more than alphabets are more than 10, so it will skip the name. So better to use the length of your names in the database. Almost is near about 50, or you have included the more information. Most probably let's choose 80, because normally it does not. So this will be expired. Okay, save. So it is already converted here. The Philip file, yeah, Philip file 31, Philip. I can don't save. So this is our file name. Copy and output. This is our file name in the Philip. Oh, this is the bundle Philip. Okay, fine. This would be our output file. Let's see. I'm going to give the name 31 output file, and this is text file where we have this information like how much is the IT, LSU, SSU, and TEF, and then just save it and save, close, and after just run. Oh, there is an error here. The model does not exist. Okay. To So previously we have this model here.
Yes, Raja, we can. Yeah.
Yeah, we can. Okay, because there is an error. Rounded the number of species and sites. Yes, Todd Philip. Yes.
Raza, do you have any previous file which is already good?
Yeah.
Just run that.
Yeah, it started. Yeah, it's running right now. So after finishing, we will get the 5 files here in the Roxmail that we have mentioned in the presentation. So we will get the output file that the name we have given in this command. And when it will finish, we will get the Roxmail tree. And after that, we have to open, let's open a figure tree because I already have that file. And then I'm going to just close when it bootstrap value is 120 and it will be to the 1000. It will, after that, it will generate the bootstrap values later, or maximum tree. Now it's bootstrap values about 1000 times. Later on, it will generate the maximum likelihood tree, best tree. And just I'm going to cancel it right now. Okay, because we already have the file. And figure tree, go here. So file, open, go to output file by partition. Yes. So this is our final tree. So we know that which one is our out group. And I remember out group, I think we have to check the out group. I can check here. So outgroup is this galenium. So this one is our outgroup. Go, reroute, tree, decreasing note order, and we have this one. So, and after tip labels, we can go to 12, font size, Arial, okay, but we can zoom it and or the root length like this, and expansion would be this. Okay, let's see. And the missing values are the branch label, branch label, branch length, label, and font size should be 10. And these are the bootstrap values we have. And this is a root node. And with the outgroup, this is our final tree that is maximum likelihood. So we can export PDF to, let's go to like this, okay. PDF format, and it's saved in the PDF format.
So, Raza, can I
So
come in?
this, yeah,
Or do we want to show them some more things today?
yeah, this is the Roxmail tree, and after that, just the command for the Mr. Base.
How long it might take? Couple of 5 minutes?
Maybe, yeah, 10 minutes. Yeah, just 10 minutes.
Okay, yeah.
Yeah. So this is the final tree that I think it's did not save. It's not opening, but we have the same result here. There is some kind of error in this. So we have these result in the PDF format. We can edit this tree in a Adobe Illustrator for make it beautiful and make it add the values and to tree edit and change the name as well as whatever we want to do some addition, but we cannot change the bootstrap and root length, everything. So now let's move to the Mr. Base. This is Roxmail. So in the backend, we have the Mr. Base, and we have our system is 64-bit. This is the file, that Nexus file we generated. Nexus because before here the Texa and the mention, and we have like generated the file already. And at the last is a command line, Mr. Base command, and we have the model test information here, the fixed values and P inverse values and shape P inverse values changed 100,000 times, and that's so I already explained these things in my book chapter as well as in the presentation how we can replace them, and these are present in a figure form. And just let's after making this file we can run 64 and command based, go there, execute. The file name is this one, Astrobarium genus, like HYSTEROB .NEXUX, Nexus file format, because now this is error. Why? Because we have this hashtag. So execute HYSTEROBIUMNEX. Oh, I think name is wrong. Execute HYSTEROBIUMNEX.
Raza, your voice is breaking, Raza.
Is okay?
Okay, now, thank you.
You'll see this is running the maximum Mr. Base. So it will take a long time to finish, and when it will finish, I already here I think have the information. Okay, let's close it. So it's running already, and it will finish. It will take some time and to get finished. And when it will finish, how it looks like? It will looks like this. We have the tree, and because I just recently finished this one, and all the information are here, and we have to execute some information to add in the phylogenetic tree, how we have used this information like probabilities or how much, so we can execute from a input information file, another file from that, so we can execute those information from there, and later on the same when just close it by this already finished. So go there, figure tree, open Mr. Base. Then we already have in the backup Mr. Base, there, Rocksmill, Mr. Base, which file? Oh, I already closed it with.
Raza, just make sure of your voice, please. Thank you.
Yeah, sure. This is MCMC open label. Oh, no, MCMC file. This is a tree file we have to open, so we have the same outgroup here as for Golini. Our outgroup, reroute, go, decreasing node value. So in the same way we will use, and then compare the topology manually. Normally, maybe if there is a software to compare the topology, of course, we have to follow now the same step: branch, node, time scale, appearance, branch tip label, name, user selection, font, area, okay, expansion, branch label, probabilities, 10, and then we can save as the PDF and then compare normally manually or maybe with the software. And that's all. Then we can compare if topology is okay, our methodology, our tree is okay, and then we can use the Rocksmill tree, and we can add these in these values in the Adobe Illustrator, or we can use some other software to add this value in one tree. That's all is about the Rocksmill and Mr. Base. Yeah, Dr. Vele, yeah, I'm okay. Now it's finished from my side.
Thank you, Dr. Raza. It has been a fantastic session of practical. I know for many of the people in the audience, it may not be so clear, maybe it might be so overwhelming. But if you refer to Raza's book chapter, so many of the things will be clarified, I guess. We will take maybe 5 questions now. Raza, Dr. Raza, how about that?
Yeah, all right.
So if the people from the audience have any questions, please ask now before we go towards the assessment quiz. Looks like there are no questions. Thank you. Raza, can you stop presenting? Yeah.
Okay, thank you.
Dr. Raza, I would like to thank you, from the bottom of my heart, for conducting this online workshop. You have been fantastic.
My pleasure.
You have been fantastic. You have worked really hard. You have worked very hard to make this kind of a very intense training. Of course, we named it advanced phylogenetic analysis. We named it for a reason, because advanced phylogenetic analysis is something not easily understandable at the beginning. It would need some kind of mentorship, and I believe Dr. Raza can be a mentor for many of the people here. I think you can contact him on his email. Maybe we will share his email address in the chat box. How about that, Dr. Raza?
Okay, all right. Maybe we can share the slides and the software if someone needed, the files they needed, I can share those software.
Thank you so much, Dr. Raza.
If they want to use the same steps from that we conducted in the presentation, that's okay. I can share those same file with them.
Yeah, thank you, Dr. Raza. Now we can let you go now. You are welcome to stay back. But we will have an assessment quiz. If the people from the audience are interested to receive a certificate of completion, you are required to attend this online quiz. Are you ready? Please type in.
Okay.
Are you ready?
Yeah.
Except for 3 responses, most of them have scored 10 or more. Congratulations to all the participants. Thank you very much. And with this, I'm gonna close down wind up this session. If you have any queries, please email. Our email address is mycoasiajournal@gmail.com. Thank you very much, ladies and gentlemen. Have a good day there. Thank you. Bye-bye.